Hi, my name is Paola Rosas. I am an assistant professor at the College of Pharmacy at the University of Illinois, Chicago. And today we just wrap up a great session in cardiac sarcomeres and cardiomyopathies, new perspectives. And we want to highlight some of these presentations. And today I have Dr. Julian Stelzer, who is a professor at the Department of Physiology and Biophysics at Case Western Reserve University. So, Dr. Stelzer, tell us a little bit more about your topic today.
Okay, well, thanks for having me. So today I spoke a little bit about our approach to cardiac myosin-binding protein C therapies in terms of really how to understand how to utilize them and how to manipulate this really exciting protein and the kind of things that you can do in the heart, both in vivo and through some biophysical measurements to try to understand mechanisms. So our approach in general has been to try to find ways to manipulate sarcomere function. So of course there's lots of myosin work going on right now. So our approach is really to look at different proteins that modulate myosin activity as opposed to myosin itself. And we think that myosin-binding protein C is one of those great targets that could really be leveraged in vivo. So basically our research is really focused on trying to understand how we can use this protein to manipulate different aspects of in vivo function. So our studies have been mostly focused at looking at different regions of the protein, trying to understand what the different parts of the protein do in terms of manipulating both contractile function in vitro and in vivo, and then trying to see if we can apply those concepts in vivo to different heart failure models. So our initial approach is really to take mouse models that have either protein depletion of myosin-binding protein C or expressing different mutations in myosin-binding protein C, so we can try to, using gene therapy approaches, express these proteins in the heart, different regions of this protein to try to really target the very specific contractile defect that these mutations have. So doing that, we can maybe create a more targeted approach to treating heart failure and actually looking more at mechanism-based therapies, which I think is really where the future of heart failure treatment is.
Thank you. And talking about gene therapy, what do you think is the future of using gene therapy to target myosin-binding protein C?
Well, so I think that in the near future, probably the most reasonable studies that need to be done is really looking at the easier mechanisms to understand in humans, which is the mutations that really cause haploinsufficiency or decreased protein expression. And of course, those are easier to handle because we know that if we can replace the protein in these patients, we can reverse really some of the contractile dysfunction and compensatory mechanisms that they have. This is based on a lot of animal work that has been done in our lab and other labs. So beyond that, I think looking at patients with more complex disease mechanisms like missense mutations might be the next step, although those are definitely challenging to decipher because there's so many with different diverse mechanisms. And I think in the long run, what the goal could be is again to use myosin-binding protein C therapies really to treat many forms of acquired heart failure by understanding how to manipulate in vivo function in specific ways that could be targeted to different diseases that have that specific defect.
Great, thank you very much, Dr. Stelzer, for this great presentation. So he highlighted the significance of myosin-binding protein C in normal cardiac function and how its dysfunction can result in heart failure. And also he emphasized on the potential of targeting myosin-binding protein C as a therapeutic option to treat heart failure. Thank you very much.
Okay, thanks for having me.